Thank you, Dr. Kavya ji. Very good afternoon to all. I'm very much pleased and honored to take part in this iconic event organized by SRIVT, marking its Silver Jubilee celebration. I'm also very much thankful to the director of SRIVT and also the organizing team, for giving me this opportunity to share my views, particularly to share some of the basic strategies towards driving international recognition of our Indian traditional knowledge. Yeah. So, my childhood days were very pleasant, and I'm very much grateful to God for that. I always, cherish the memories. One of the cherished memories, include the drinking of kashayam. So, most of you may be aware of this kashayam, which is a traditional Siddha formulation, made out of herbs. So every Sunday morning, without fail, my mom used to give me this kashayam made out of several herbs like neem shoots and then dry ginger or vasambu and nutmeg, all these. So before I got introduced to these medicinal plants by learning the course pharmacognosy or advanced pharmacognosy during my bachelor's program, I just got introduced, or gained some knowledge about these medicinal plants through my mother and my grandparents. So this, kindled the interest inside me to do a research in the area of medicinal plants, particularly to know about the chemical constituents, that are responsible for the activity. Because parents, when they are giving the food, or any kind of herb or leafy vegetables, they say that, see, if you eat this, it will give you this benefit. But how does it give the benefit? What are the components that are present, and that are responsible for this activity? That is not, properly clarified to a kid. Yeah. So the Indian traditional knowledge, I strongly believe in this, and it has gained the popularity worldwide. But still, you can see that it has not attained the complete acceptability as it of the allopathic medicines. Because allopathic medicines, are considered to be well regulated, and there is a very stringent regulation on controlling the quality and the safety of those medicines. So when the Ayush products, okay, or dealt, people say that they are not very well regulated products, particularly about the chemical constituents, or the formula are not well clarified. So now when I have always wondered that, why, even though, the Ayush products, have gained the popularity, but why it has not gained the international acceptability? That's again, was a question, that was bothering me. I have referred to the literature and also discussed about this with the experts. So what I understood is, my view is, if we give a scientific validation, okay, or a scientific evidence for the activity, of the Ayush product, or a scientific proof for the traditional claim, it will be acceptable. So we have to build, So the traditional knowledge, has to join hands with advanced technologies, okay, to build the trust. And this will give the international recognition, right? So through this, we are going to give a patient safety. And always, people ask that, of course, medicines, are for patient safety, scientific evidences, why do we have to bring out proof, okay, or a scientific validation using these advanced technologies? Because it's going to give the identity for the medicine. Because there are several herbs, which have very synonymous names, okay, the regional names are very much similar, but what is that exact herb that is used, for making the formulation, that need to be identified clearly, and also to get the consistent quality of the medicine, okay, not to have any kind of batch to batch variation, during the Ayurvedic or any Ayush products manufacturing, okay, so we have to have a control. So for this also we have to use the modern techniques, okay, to give a consistent quality medicine, and also particularly to make sure that there is no toxicity of these herbs that are used in making these formulation. Yeah, so safety and efficacy of the medicines are very important, okay, and regulatory requirements, conforming to the regulatory requirement is again very important, okay. There are European regulatory agencies, and also recently, CDSCO, our regulatory body, has also come out with certain improved regulations, okay, for the Ayurvedic or the Indian traditional products, okay. So we have to conform to that. For this, again, we have to give the proof. Yeah. So for the overall, improvement of the health and the well-being, okay. So we have to work together, to bring the scientific evidence. Yeah, so there are some official resource materials published by the Ministry of Ayush. Yeah, so the Ayurvedic Pharmacopoeia of India, for the formulations and for the herbs, they have published. So they have set certain set of standards, okay. They have prescribed the standards, that ought to be complied by any medicine that is manufactured by the Ayurvedic manufacturers. But according to the regulatory guidelines and pharmacopoeias, if you see, identification is majorly based on the macroscopic and the microscopic evaluation, and recently they have also included the chemical profiling of the herbal materials. So with this, they are assuring that the quality and the quality of the Ayush projects, okay, or products, okay, could be controlled, and the standardization, okay, could be achieved. Sorry. Yeah. So what are the major challenges, okay, related to these medicinal plants or the herbal preparations? So there are variable sources, for the herbs and their chemical complexity. And we need to make a right identification of the herb, and we have to have a quality assurance of the starting materials that are used, for making the preparations. So that's going to contribute to the safety and the efficacy of the final product that we'll make. So this is again going to validate the traditional knowledge. So all that we have to do is the quality control and then the standardization using modern analytical and molecular techniques. So this is going to give the scientific validation for the safety and the efficacy of the herbal extracts. So the point which I want to insist on, okay, with this speech is development of the markers. So using a marker compound, particularly the bioactive substances as markers for the standardization of the extracts is very, very important, okay, if we want to make our market, okay, internationalize. So there are different types of markers that are used by companies, okay. So DNA markers are used, and this can be extracted from the fresh or the dried plant tissue, and this can be used for identifying the genetic variation, medicinal plant breeding, and also for the authentication of the medicinal plants. And the second type, biochemical markers, these are proteins or the enzymes. This again can be the enzyme pattern, okay, can be used to distinguish the closely related species, okay, and to identify the specific species, that you want to use it as a raw material for your herbal formulation. And chemical markers, so authentication of for the authentication of the genuine species, yeah, so you have to use a chemical marker, okay, and these markers again are useful to come out with the substitutes for the herbal materials also, because there are many species which are found to be endangered, okay. So if you want to bring a substituent, okay, to any of the herbs, okay, then definitely you need to have a chemical marker, okay, to prove the activity, okay, or the bio equivalent activity, okay, of the herb. Yeah. So I will just describe, the general procedure, how we can develop these chemical markers, okay, and you can use them for the standardization purpose. Yeah. So it's not an easy task, to isolate or purify the individual constituents, from the complex extract, okay, and to develop them as the chemical markers, right? So there are several extraction procedures that are in use, okay. So you can use either a solvent extraction method or a Soxhlet based extraction method or microwave assisted extraction method or ultrasound assisted techniques or microwave supercritical fluid extraction, or a simple distillation or the hydrodistillation method could also be used, for extract preparation. And when the crude extracts are prepared, then you can take it forward for the enrichment process. Yeah, that again can be done with the help of the solvent partitioning method or the fractionation method or adsorption chromatography you can use, or membrane filtration techniques can also be used, okay, or solid phase extraction methods can also be used for this purpose. So now when you want to develop an marker, you have to isolate the individual components, particularly the one that has the biological activity. So for that, we scientists are majorly dependent on the various chromatographic techniques, like preparative HPLC method or the high-speed countercurrent techniques can be used, or simple sometimes, if a plant has a particular constituent, okay, in major amount, then a simple crystallization method would also be helpful. Yeah. So to get a very pure molecule, sometimes you may have to go for the multiple times, doing the chromatography. Yeah. So if you are focusing on to the bioactive components, then it is advisable to use the bioassay guided procedure for the isolation and the purification purpose. Yeah. So you can have the extract that is prepared using a range of solvent, yeah, from the non-polar to the polar you can have, and then you can subject it to some simple cell-based assays, okay, so to identify, which solvent-based extract, okay, is found to have the activity. So once when you have identified that, then you can go for doing the fractionation using different solvents, and all these fractions can again be subjected to a bioassay. Yeah, and then identify the specific isolate or the fraction, that is found to have the activity. So now
Madam,
only
you have
this fraction,
5 more minutes left.
yeah, only this fraction, you can take it forward, for the chromatographic methods. So this is again going to have the bioactive substances, and these bioactive substances, isolated, can be subjected to various structure elucidation methods and can be further developed as the marker molecules. Yeah. So there are several structure elucidation techniques, okay, that are known and that can be used. So traditional ways or the chemical methods, okay, and the modern ways, you can for the modern ways, you can use various spectroscopic methods, okay. So UV IR spectroscopy, proton NMR and the carbon NMR techniques, okay, can be used, and there are some advanced nuclear magnetic resonance techniques or used, okay. So one dimensional and the two dimensional techniques, okay, are available, and that you can use for the complete structure characterization or the structure elucidation of the molecules. Yeah. So in the pharmacopoeia there are some complete information. They say it is complete information, but in some aspects, the information or the standards, prescribed in the pharmacopoeia are found to be incomplete. Okay, this is again my personal view. Yeah. So about the complete information about the therapeutic uses of the drug, okay, and identity, purity, and then strength, all the information and the limitations are prescribed in the pharmacopoeia. So here I have taken one example, that is Bringraja. So very popular Ayurvedic plant. Yeah. Available as a single drug-based single herb-based formulation, or multi-herb-based formulation. So prescribed for various therapeutic uses, okay, as mentioned here. So the point I want to insist on here is, so the identity, is recommended based on the TLC method and also based on the HPLC method. But if you carefully look at the HPLC chromatograms that are presented on the right side, the figure A, B, and C, okay, if you see there carefully, yeah. So it's only that one compound that is present in this extract, okay, has been identified and quantified, which is vidilolactone. But if you look at the chromatogram, there are multiple peaks in it, which comes as an evidence that this extract contains other constituents also, but which are not characterized. So although the limit is set based on the one component, that is vidilolactone, this extract contains several other components, okay, which might be contributing to the activity and also the toxicity, isn't it? So those need to be again quantified. I have one more example, okay, that is the extract of this Solanum suritans or the xanthocarpum, which is yellow berried nightshade fruit. The whole plant again is recommended, okay, for various therapeutic uses as mentioned here. So
Madam,
here
please
also,
wind up, madam.
yeah, sure. So here also you can look at the HPLC chromatogram. This is just an extract from the Ayurvedic Pharmacopoeia, okay, reference I have given below. So you can see here again the identification has been done, okay, for only one constituent, okay, that is solosodin, yeah. But the fingerprint, the HPLC fingerprint, if you see, there are several components, they were not at all characterized. Yeah, that would again give a significant contribution to the activity and the toxicity, right? So we have to bring out the scientific evidence, okay, for the presence of the major constituents, okay, at least the major constituents I'm saying. So if you look at the chromatogram, there are peaks, which have the intensity very much equal to that of or more than that of the solacodin, right? But those have not been characterized. So those are some lacunae, I would say, those again can be covered, by using some modern techniques. So here I have a case study, where the standardization of a single herb based formulation, okay, that is CLIV 92, and the standardization has been done, okay, based on the bioactive components Clium viscosins A, B, and C. So how the characterization of this Clium viscosin A was done using various spectroscopic methods. As there is shortage of time, okay, I will not explain, how we have done it, but at least the techniques, that were used for the the characterization of the complete structure, okay, that I will say. Please allow me a few minutes time. Yeah. So using UV absorption spectrum, we identified the presence of two major chromophores, that is the substituted coumarin and substituted benzene. Yeah, and from the IR spectrum, we identified the presence of the phenolic OH group and then the lactone carbonyls and the aromatic functionalities. And using EAMS, we could able to determine the molecular formula. Yeah, and we went for measuring the proton NMR spectrum, okay, and the carbon NMR spectrum, okay, to know about the types of carbons that are present, okay, and also the different types of hydrogens that are present in the molecule. Yeah, so we got the proof, for the presence of the coumarin nucleus, okay, and the aromatic nucleus, and also the oxymethylene protons, but we could not get the proof, for the oxygens, okay, that we could identify from the mass data, okay. So we could go for doing some simple chemical conversions, okay, to confirm the presence of certain functional groups, okay, and we could arrive at determining the part structure of the compound, and a simple double bond equivalence calculation helped us in identifying the presence of an additional ring, okay, that connected this coumarin and the phenylpropane nucleus. So I will skip. Yeah, so and the presence of the dioxine bridge again was confirmed, based on the mass spectroscopic analysis. the various fragmentation, and this compound, we could isolate in good quantity, so we could do some chemical degradation also to determine the complete structure. So once based on the spectroscopic techniques, we identified the groups and then we built the structure. We finally, had a confusion whether the isomer A is the structure or the isomer B is the structure. To resolve this, we went for carrying out the two-dimensional NMR analysis, that is the heteronuclear decoupling experiments. So irradiating some specific functional group and with the sharpening of certain signals, we could identify the linkage and we could determine the structure. So we can use all the modern spectroscopic techniques, to characterize the structure and to develop, and we can develop the markers. So this will again be helpful for the identification of the adulterants and for the differentiation of the herbal medicines with the multiple sources, and also for the determination of the best harvesting time, and also for the confirmation of the collection sites, and even to optimize the processing methods, and also to well hold the quality of the herbal product, particularly in terms of the stability, and then the identity, its concentration. So everything we can do with the help of this, and we can even link this traditional knowledge to the natural products drug discovery. We can discover drug lead molecules, and we can even develop them into an allopathic medicine also, right? So phytopharmaceuticals, recently approved a type of drug by our Indian regulatory agency. So this represents a fascinating intersection between the traditional knowledge and the modern science advancements. So it is nothing but a purified standard fraction with defined number of the phytochemical compounds. Minimum of 4 components need to be quantified, identified and quantified, and prove that they were the components that are responsible for the activity of this phytopharmaceutical. Then you can get an approval from CDSCO, and you can market your product. And so here I would like to
ma'am, please, we are really running out of
time.
Done.
Yeah. Done. Thank you. So these are some of the novel compounds that were discovered from our lab, only from the plants I have presented here. And in our lab, we are even working on exploring the bioactive molecules from various natural sources, from the microorganisms and also from the marine sources also. We are trying to explore the drug lead molecules. So I'm not going in detail to it. Yeah, so we have all the facilities, to develop a chemical marker. So in this forum, there may be some startup company, or some researcher who will be looking forward for a researcher, to collaborate, particularly for the standardization of the herbal extracts. Again, herbal extracts, or complex materials, will definitely have a mixture of components, but we have all the facilities, in our institute. If anybody is interested, you can contact us. I have given here my contact ID. Thank you very much for your patience. Sorry.
Thank you, Dr. Sajeli Begum Garu, for your insightful speech. I would like to ask Session Chair Dr. Kavya Dashora Garu to present memento, shawl and certificate to our speaker.